Good afternoon. Last day, we had discussed about the excitation, contraction, coupling of the skeletal muscle and a bit also about the cardiac muscle. Let us just revise about the structural organization of the muscle. That is, each muscle fiber or myofibril is surrounded by endomysium. The muscle fibers are grouped together to form fasciculi, and each fasciculus is surrounded by perimysium. All fasciculi, they collectively form the muscle belly, which is surrounded by the epimysium. At the junction of the muscle with its tendon, the fibers of endomysium, epimysium, perimysium become continuous with the fibers of the tendon. The tendons are fibrous terminal ends of the muscle made up of collagen tissue. So we have discussed about this. Then we have said that the structural and functional unit of the muscle is the sarcomere, which is the portion between the two Z lines. The T-tubules in cardiac muscles are more well developed as compared to the skeletal muscle, while the sarcoplasmic reticulum, it is very well developed in the skeletal muscle. The terminal cisterns of the sarcoplasmic reticulum along with the T-tubule form structures called triad, while in the cardiac muscle they form a dyad. So now let us discuss about load, tension, optimum length, equilibrium length, work done, and the Frank-Stirling's law. So what is load? Load is the force exerted by the weight of an object on the muscle. So load is the force exerted by the weight of an object on the muscle. Now let's see about tension. The force exerted by the contracting muscle on the object is known as muscle tension. So, muscle load and muscle tension are two opposing forces. The load acting on the muscle can be of two types. One is free load or preload, and other is afterload. A load which starts acting on a muscle before it starts to contract is called free load. Let's cite an example. In an intact body, say a muscle example is filling water from a tap by holding a bucket in the hand. The free load increases the force of contraction and work efficiency of the muscle. The free load stretches the muscle passively, producing a passive tension across the muscle. This passive tension increases the force of muscle contraction in two ways: by increasing the initial length of the muscle, by adding an elastic recoil force to the muscle during its contraction. So we have seen about load and tension. Resting length, it is the length of the muscle at rest. Optimum length is the length where maximum active tension is built up in the muscle. Equilibrium length, say you take out a muscle from the body to do some experiment. You detach the muscle from its bony attachment. So the length the muscle adapts now is called the equilibrium length. And work done, you know, is equal to force into displacement. Now we should know something called the Frank-Stirling's law. Greater the initial length of the muscle fiber, greater will be the force of contraction within physiological limit. Last day, you have seen how a muscle contracts, the molecular basis of muscle contraction. There is cross bridges which are formed due to interaction between the actin and myosin filament. At optimum length, there will be maximum number of cross bridges that will be formed. Now, outside physiological limit, what will happen? The myosin and actin filament will move away from one another. So the interaction will decrease. We will explain this later on also. Now let's discuss about different types of contraction that occurs in the muscle. One is isotonic contraction, another is isometric contraction. So isotonic contraction, here the tension in the muscle remains constant, but the length of the muscle changes. So in isotonic contraction, the tension in the muscle remains constant while the length of the muscle changes. In isometric contraction, the length of the muscle remains constant while the tension in the muscle changes. So isometric and isotonic contraction. Now, isotonic contraction can be of two types. One is concentric contraction, another is eccentric contraction. So in this diagram, you can see in isometric contraction and isotonic contraction. So in isometric contraction, there will be no work done because there is no change in length of the muscle. But in isotonic contraction, there will be change in length of the muscle. So work will be done. Now, there are different components in the muscle. One is parallel elastic component, another is the series elastic component, another is the contractile component. So contractile component, parallel elastic component, and the series elastic component. In an isometric contraction, you can see this is an isometric contraction. This A and B, the pictures, you compare them. You can see what is happening here. The total length of the muscle is remaining constant. So this is isometric contraction. What happens here? The series elastic component, you can see here the series elastic component. This is the series elastic component. It is stretched. Though the contractile component, it has shortened as compared to A, you compare A with B, you can see that the contractile component, they have shortened, but as the series elastic component, they have been stretched, so the total length of the muscle remains constant. So this is isometric contraction. What happens in an isotonic contraction? Here you see C and D. Here, the series elastic component is not stretched that much. So what happens? The contractile component, it has contracted. As a result, the length of the muscle has decreased, but the parallel elastic component, it is folded to absorb the change in tension in the muscle. So the tension in the muscle in this case remains constant. Isometric exercise, you can see power games like sprinting, weightlifting, boxing. These are the examples. Isotonic exercise. In this kind of exercise, majority of the muscles are in metabolic state similar to isotonic contraction. Energy is derived by the muscle through aerobic metabolism. Isotonic exercises lead to increasing endurance of muscle. Target muscles are those with predominantly slow twitch red fibers like the soleus muscle. Isotonic exercises, marathon running, swimming, cycling. You can see the pictures. So these are isotonic exercises. So in isotonic exercise, we have said that the tension in the muscle remains same, but the length of the muscle changes. there are two types of isotonic contraction. We have told one is concentric, another is eccentric. Concentric contraction occurs when the muscle shortens. You can see here, this was the muscle. Here it has moved towards the joint, so there is shortening. So this is concentric contraction. Now eccentric contraction. This was the initial position. Now you have stretched the elbow, so the muscle increases in length. This is eccentric contraction. So concentric contraction and eccentric contraction. Isometric contraction, we have told that the length of the muscle remains constant. So you can see here, length of the muscle is remaining constant. That is, no movement is occurring here. So again, you can see isotonic contraction, isotonic contraction. This is an example of an isometric contraction. Isometric contraction, isotonic contraction. So in case of isometric contraction, there is no change in length. So work done will be 0. Now let's see about the length and tension that is developed in a muscle. Here you can see this is the sarcomere, that is the portion between two Z lines. Say this is half the length of the muscle, optimum length of the muscle. In this case, what is happening? There is excess overlapping of the fibers, so less interaction of the myosin and actin. The tension developed in this case is purely active tension, but it is not the optimum length of the muscle, but active tension is built up in the muscle. In B, you can see this is the optimum length of the muscle. Say this is the optimum length of the muscle. What happens here? There are maximum number of cross bridges that are formed. So active tension built up in the muscle is maximum. Now what is happening? Now you are stretching the muscle more. You are stretching the muscle more. So there is interaction between the muscle, but the actin and myosin have started moving away from one another. So the number of cross bridges are decreasing. The tension in the muscle is increasing, the total tension, but the active tension in the muscle decreases. Now the point has come D, that is outside the physiological limit. You can see here the actin and the myosin filament, they have moved away from one another. As a result, the tension built up in this case is passive tension and not the active tension. The total tension has increased, or then that is only contributed by the passive tension. So here you can see we have plotted the tension in the muscle. Here is half the length, normal, that is half the optimum length. This is the optimum length of the muscle. This is twice the optimal length. So what is happening? This is half the length. So the active tension, now you can see the active tension is increasing after the optimum length of the muscle. Now what is happening then onwards? The active tension is gradually decreasing when you are increasing the length of the muscle. A time comes when the active tension will be further decreased and the passive tension will be built up in the muscle. So this is, here we plot the velocity of contraction. This is the load opposing the velocity of contraction. So you can see the load and the velocity of contraction, their relationship in this curve. So skeletal muscles are the principal executor of exercise. Human skeletal muscle can exert maximal contractile force of around 3 to 4 kg per square centimeter of cross-sectional area. So this is called the strength of the muscle. The strength of the muscle is determined mainly by its size, with a maximal contractile force between 3 and 4 kg per centimeter square of muscle cross-section area. So that is the strength of the muscle, strength of the muscle. muscle power is generally measured in kilogram meters per minute. Kilogram meters per minute. Power is determined not only by the strength of the muscle contraction, but also by its distance of contraction and the number of times that it contracts each minute. So the power of the muscle contraction is different from the strength, because power is a measure of the total amount of work that the muscle performs in unit period of time. So power of the muscle and strength of the muscle are somewhat different. Now let's see about the energetics of muscle contraction. So the ATP that is stored in the body is only able to provide you energy for 1 to 2 seconds. Then comes the phosphocreatinine system or the phosphagen system, which can provide energy for 8 to 10 seconds, 8 to 10 seconds at a rate of 4 moles of ATP per minute, at a rate of 4 moles of ATP per minute. Then comes the glycogen lactic acid system, which can provide energy for 1.3 to 1.6 minutes at a rate of 2.5 moles of ATP per minute. Then comes your aerobic system, which can provide energy for unlimited time, that is as long as nutrients last, at a rate of 1 mole of ATP per minute. So this is how you get energy when you do some exercise. Another measure of muscle performance is endurance. This greatly depends on the nutritive support for the muscle, more than anything else on the amount of glycogen that has been stored in the muscle before the period of exercise. So that is called the endurance of the muscle. So you have learned about the strength of the muscle, the power of the muscle, the endurance, and the energy where it gets, the muscle gets during exercise. You have learned about the phosphocreatinine system, the lactic acid system, then the oxidative metabolism. Now we will see something called oxygen debt. The body normally contains about 2 liters of stored oxygen that can be used for aerobic metabolism even without oxygen. So in your store there is 2 liters of oxygen. So how is this 2 liters stored? Let's see about that. 0.5 liter in the air of the lungs, 0.25 liters dissolved in body fluids, 1 liter combined with hemoglobin of the blood, and 0.3 liters stored in the muscle fiber themselves, combined mainly with myoglobin. Now we are doing a heavy exercise. So all these are used within a minute or so. So where will you get the rest of the energy? So you are doing an exercise, heavy exercise for 15 minutes. So you need some extra energy. So the extra energy you will get by using the different system we have discussed till now. First is your stored ATP, followed by the phosphocreatinine system followed by lactic acid metabolism. So they will all provide you with energy. So after you do the exercise, you need to repay the debt that you have incurred by using these systems. The debt is the oxygen debt. It is 11.5 liters. 11.5 liters. The early portion of the oxygen debt is called the lactic acid oxygen debt, and it amounts to 3.5 liters. The later portion is called the lactic acid oxygen debt, and it amounts to 8 liters. So even after doing an exercise, what you will need to do, you need to consume more oxygen for a certain long time of interval, say 30 minutes, 40 minutes, to repay this debt that you have incurred during exercise. So the debt you have incurred is 11.5 liters, among which 3.5 liters is your lactic acid oxygen debt, and the rest, that is 8 liters, is your lactic acid oxygen debt. So we have learned that how 2 liters of oxygen is present in your body. You are using them. There are different parts. Here you can see them. And here you can see the lactic acid and the lactic acid oxygen debt. 8 liters and 3.5 liters, which need to be repaid. So we will now discuss about the heat production in muscle. Thermodynamically, the energy supplied to a muscle must equal to energy output. The energy output appears in work done by the muscle, in energy-rich phosphate bonds formed for later use, and in heat. The overall mechanical efficiency of skeletal muscle ranges up to 50% while lifting a weight during isotonic contraction, and it is 0% during isometric contraction. Energy storage in phosphate bonds is a small factor. Consequently, heat production is considerable. The heat produced in muscle can be measured accurately with suitable thermocouples. Now, there are different heat. One is resting heat. Resting heat is the heat given off at rest. Is the external manifestation of basal metabolic process that is occurring in the muscle. The heat produced in excess of resting heat during contraction is called the initial heat. So you have learned about resting heat and initial heat. This initial heat is made up of activation heat, the heat that the muscle produces whenever it is contracting, and the shortening heat, which is proportional in amount to the distance the muscle shortens. Shortening heat is due to some changes in the structure of muscle during shortening. So we have learned about resting heat, initial heat, activation heat, shortening heat. Now we will see about recovery heat and the relaxation heat. Following contraction, heat production in excess of resting heat continues for as long as 30 minutes. This recovery heat is the heat liberated by the metabolic processes that restore the muscle to its contraction state. The recovery heat of muscle is approximately equal to the initial heat. That is, the heat produced during recovery is equal to the heat produced during contraction. If a muscle that has contracted isotonically is restored to its previous length, extra heat in addition to recovery heat is produced. That is the relaxation heat. External work must be done on the muscle to return it to its previous length. And relaxation heat is mainly a manifestation of this work. So we have learned about resting heat, initial heat, activation heat, shortening heat, recovery heat, and relaxation heat of the muscle. We have seen about the tension in the muscle, that is the force that the load exerts on the muscle. That is the tension of the muscle. Passive tension, it refers to tension developed by the parallel elastic component, also known as non-contractile tension. Active tension, it is mainly due to the interaction between the contractile components of the muscle, that is the actin and myosin filament, the thick and the thin filaments that are present in the muscle, that cross bridge formation due to them. You have learned that a motor unit is a single motor neuron and the muscle fiber it innervates. They form the motor unit. Each spinal motor neuron innervates only one kind of muscle fiber. So all the muscle fibers in a motor unit are of same type. On the basis of the type of muscle fiber, they are innervated, and thus on the basis of the duration of their twitch contraction, motor units are divided into slow, fast, resistant to fatigue, and fast fatigable units. There is also a gradation of innervation of these fibers. So let's see how is this gradation. With S fibers tending to have low innervation ratio, that is, they are small units. Fast fatigable fibers tending to have the highest innervation ratio, that is, they are large unit. The recruitment of motor units during muscle contraction is not random. It follows a scheme, the size principle. A specific muscle action is developed first by recruitment of S muscle units that contract relatively slowly to produce controlled contraction. Next comes the fast fiber, which are resistant to fatigue. Lastly, fast fatigable fibers are recruited for more demanding tasks. Say you are standing. So slow fibers are recruited. Now you have started walking in slow motion. So fast fibers, but resistant to fatigue, they are recruited. Now you start running and jumping. So fast and fatigable fibers are recruited now. This is the size principle. This is the size principle. So we have learned about isometric length tension relationship already. We have seen the optimum length where the maximum actin myosin interaction occurs. As a result, the active tension buildup is maximum here. This is the, we can see in this picture, resting length, optimum length, active tension, passive tension, and total tension. So if you increase the length of the muscle beyond the optimum length, the passive tension will increase. And if you increase the length of the muscle beyond the physiological limit, there will be no more interaction between the actin and myosin filaments. As a result, there will be force of contraction will not increase any further. So this can, with this you can explain the Frank-Stirling's law. More the initial length of the muscle fiber, greater will be the force of contraction within physiological limit. Now let's see about application of length tension relationship when applying to muscle joint system. Sarcomere length is not same throughout. Muscles have diminished ability to produce isometric contractions at extremes of joint motion. Torque of muscle reduces when full ROM is attempted at all joints crossed by multi-joint muscle. This decrease in torque is called as active insufficiency. Now we come to force-velocity relationship. Another factor affecting tension development is speed of shortening of myofilaments, rate at which myofilaments slide or form and reform cross bridges. Speed of shortening depends on type and length of the muscle fiber. The force generated is the function of velocity of muscle contraction. Concentric contraction, as speed decreases, tension increases. Isometric contraction, speed of shortening is 0, but tension reduced is more than concentric. Eccentric contraction, as speed of lengthening increases, tension increases, and then it plateaus. You can see here the relationship between muscle force and contractile velocity, the relation between them. Now, isometric contraction, isotonic contraction, concentric contraction, eccentric contraction, we have already learned about them. In the isotonic contraction, there is change in length of the muscle. So work is done. In isometric contraction, there is no change in length of the muscle, but the tension in the muscle changes. So no work is done in isometric exercises. Motor unit, a single nerve fiber, and the number of muscle fiber is supplies is the motor unit. We have seen 3 types of motor unit. One are the small units or the slow units, another are fast resistant to fatigue units, and then the other one is fast fatigable unit. We have seen that when you are standing, what is happening? First, the slow fibers, they are recruited, followed by the fast and resistant to fatigue fiber. You have started walking, now you have started running, fast fatigable fibers are recruited. This is known as the size principle. Size principle. You can see motor units in this picture. So let us see about the working model of the muscle. Muscle consists of 3 components. One is the contractile component. Next is the series elastic component and the parallel elastic component. So you can see in this picture, series elastic component, contractile component, and the parallel elastic component. Series elastic component, tendon, parallel elastic component, non-contractile and connective tissue in the muscle, and contractile component at the actin myosin filaments. We know greater the initial length of the muscle fiber, greater will be the force of contraction within physiological limits. That is the Stirling's law. So the preload starts acting on the muscle before muscle has its contraction. So the initial length of the muscle is increased. Work done will be more. See here, initial length and preload. You can see there is a transducer and there is movement up and down to change the preload. That is the initial length of muscle. Passive tension is the tension developed by simply stretching a muscle to different lengths. Think of a rubber band. Passive tension is determined by preload and the elasticity of the muscle itself. Total tension is the tension developed when a muscle is stimulated to contract at different preload. It is the sum of active tension developed by the contractile elements of the sarcomere and the passive tension caused by stretching of the muscle. Now, length tension relationship. Muscle length is held constant at various lengths and muscle directly stimulated at many points. The develop is measured using a transducer and it is plotted in the graph. Muscle tension at rest when muscle is stretched. Passive tension is developed due to stretching of the elastic elements. Fiber using all patterns of measure. Tension is maximal, active tension maximum at 2 to 2.5 on where of optimum. Now let's see about the slow and fast twitch fibers. We have told that the skeletal muscles can be divided into slow and fast twitch fibers. Differences due to different myosin ATPase isoenzymes that are slow or fast. Red muscle fibers and white muscle fibers. Red muscle fibers are basically slow twitch fibers and the white muscle fibers, they are fast twitch fibers. There are plenty of myoglobin present in this red muscle fibers and here the myoglobin content is much less and this mainly follows aerobic metabolism, the slow fibers. Now let's see the differences. Diameter of the slow fibers small. In the fast oxidative fibers, these are intermediate in size and the fast glycolytic easily fatigable fibers are large in size. Their slow fibers are highly resistant to fatigue. Here the resistance is intermediate. Here these are low resistance to fatigue. Respiration here is mainly aerobic in the slow fibers. Here there is aerobic metabolism still in the fast oxidative fibers and fast glycolytic, they follow anaerobic metabolism. Oxidative capacity is high in slow fibers, while here in the fast fibers, oxidative variety it is somewhat high and low in the glycolytic fibers. Twitch rate in the slow fibers is slow, while fast in rest too. Myosin ATP is contained low in the slow fibers, while it is high in fibers. Now, with electromyography, during a normal muscle twitch, multiple electrical potential is dissipated into surroundings. These can be picked up by surface electrodes on the skin. And the procedure is electromyography. Electromyograph is a high gain amplifier. Keen electrodes and needles are used. Motor unit potential are displaced on CRO. Potential is a sharp spike, usually it is biphasic. Larger the motor unit potential, larger is the motor unit. Useful for distinguishing nerve from muscle diseases. EMG are obtained at rest, during slight muscle contraction, and during maximal muscle activity. Fibrillation is contraction of single muscle cells, and fasciculation is contraction of groups of muscle cells supplied by a single axon. Now let's see about the properties of skeletal muscle. Excitability. It is the change in potential and the consequent responses inherent to the tissues in response to a stimulus. Stimulus, it is the change in the external environment bringing about excitation in an excitable tissue. Stimulus can be of different types: electrical, mechanical, chemical, thermal, electromagnetic. Now, types of stimulus or the strength of stimulus can be different. One is subminimal, minimal or threshold stimulus, submaximal, maximal, and supramaximal stimulus. Now, this is the strength duration curve. We will discuss about it later. Rheobasic current, chronaxy, and utilization time. Now, rheobas. Rheobas is the minimum strength of current required to stimulate a tissue. Utilization time, it is the time a tissue takes to respond to a rheobasic current. And the chronaxy, when you increase the strength of the current to twice the rheobas, the time taken by the tissue to respond to that current twice the rheobas is the chronaxy. So chronaxy, rheobas, and utilization time. Now another property of skeletal muscle is contractility. We know there are two types of contraction. One is the isotonic contraction, another is the isometric contraction. Now let's see. You give a stimulus to a muscle, then what happens? There is a simple muscle twitch. If the stimulus is threshold, then the muscle responds. But if the stimulus is subthreshold, then there is no response in tissue that will. And in this diagram, you see that this is the contraction begins 2 milliseconds after the electrical response. So the mechanical response in the skeletal muscle begins 2 milliseconds after the electrical response. How it is, why it is like that? This is a simple muscle twitch. So you have given a stimulus. After that, there is a latent period after which the muscle starts its contraction. So this is the point of contraction. Then there is the height of contraction, follow which the muscle relaxes. After that, point G, you can see there is a curve that is known as a shutter curve or physiological curve. This is due to the inertia of the instrument. And below this graph, you can see there are some time tracing out here. This one peak and this is one trough. One peak and one trough. And with that, you can calculate the duration of different phases. This you can do the tracing with a time marker. You can see here the simple muscle twitch. First is the point of stimulus followed by latent period, then the phase of contraction and phase of relaxation. again see in this picture the contractile response. This is the electrical response. This is the mechanical response. So it begins 2 milliseconds after the electrical response. And here lies your latent period. Then comes the phase of contraction and followed by the phase of relaxation. You know, in the muscle, what happens? First, in the neuromuscular junction, an action potential arrives in the presynaptic terminal. Then there is release of neurotransmitter from the neurotransmitter vesicles, which acts on the postsynaptic membrane, and there is opening of nicotinic acetylcholine-gated receptor channels. As a result, the membrane depolarization and there is spread of action potential around the membrane. Now, this action potential travels down the T tubule where there are dihydropyridine receptor channels, which are voltage-gated calcium channels, and there is a conformational change in these channels, which causes again a mechanosensitive ryanodine receptor channel present in the sarcoplasmic reticulum terminal cisterns to open, and there is pouring of calcium from the sarcoplasmic reticulum into the sarcoplasm. This calcium now binds with troponin C and causes the troponin tropomyosin complex to move deeper into the groove of the actin filament. So now the active sites of the actin filament are free and myosin head goes and attaches to the active site of the actin filament. And now begins the contraction process. So you see there is some time gap in between these phases. The excitation followed by contraction, the excitation, contraction, which responds delay of 2 milliseconds. But in a cardiac muscle, there is the typical neuromuscular junction is not present, while there are gap junction or low resistance bridges. So the contractile response and the action potential here at the same time, while in a skeletal muscle, it begins 2 milliseconds after. You see here, this is the action potential. Here you see the contractile response. So it begins 2 milliseconds after that. So time taken by the process is action potential to spread around the T tubule, release of calcium, calcium attaching to troponin C, power stroke, calcium reuptake by the sarcoplasm. All these take some time. Now, let's see about the strength of stimulus. Here you have applied a threshold current, so the tissue has started responding. Now you increase the strength of the stimulus or the current. What happens? You can see the tissue is responding and the height of contraction is gradually increasing. The force of contraction is also increasing. But at maximal stimulus, what happens? All motor units are recruited. Here, a few motor units are recruited. Now, more and more motor units are recruited out here. Ultimately, all the motor units have been recruited at maximal stimulus. So, if you increase the strength of the stimulus further to supramaximal level, no more motor units are available for recruitment. So, the strength of contraction will not, or the force of contraction will not increase further. Now, let's see about the, if you give successive stimulus or multiple stimulus, what happens? Effect of two successive stimulus. One is beneficial effect, superimposition and summation. And effect of multiple stimuli, we will see clonus, titinus, staphy, and fatigue. And lastly, we will see the effect of temperature. Now, first, if you give a stimulus within the latent period of the simple muscle twitch, what will happen? In the first half of the latent period is absolutely refractory period. So, however the strength of stimulus you increase, you will see the height of contraction will remain constant. Now you increase, now you give the second stimulus during the second half of the latent period. You will see there is an increase in height of contraction. This is again due to an effect called the beneficial effect. If you give another stimulus during the contraction phase, you will see that the height of contraction further increases. This is again due to the process called summation. And here comes the beneficial effect. Now, what are the causes of the beneficial effect? There is, when you give one stimulus after another, there is accumulation of calcium, decrease in viscosity of the muscle, increase in hydrogen ion concentration, increase in temperature. All these are responsible for the beneficial effect. When the second stimulus is applied during relaxation phase of the curve, due to the first stimulus, the relaxation phase is cut short. The relaxation phase is cut short. You can see in this picture, here the relaxation phase is cut short. And what happens? You see the relaxation phase is cut short. And the effect of the second stimulus, you can see the height of contraction has increased. This is known as superimposition, or incomplete summation of waves. The height of the second wave is greater than the first one. This is again due to beneficial effect. Now, if you give the second stimulus just after the contraction and the relaxation phases are over, what will happen? You will see that there are two waves, one followed by the other, but the second wave, the height is more. This is again due to the beneficial effect. Now we will see the second contraction develops a greater tension from the first one. If the second stimulus is applied before the relaxation is complete in the first one. This is due to increase in availability of more calcium. Calcium from the first contraction is not pumped back by the Sarka pump and it is available. Second stimulus also releases calcium and adds to the remaining calcium from the first contraction. Now we will come to effect of multiple stimuli. So you are giving one stimulus after other. Multiple number of times stimulus you are giving. So if you give the stimulus just after the relaxation is over, what will happen? You will see the next contraction is higher than the first one. And you will see a phenomena called, you can see there is rise in the height of the waves. You can see here there is rise in the height. This is called the staircase phenomena or trippy. In this part, you are giving the stimulus during the relaxation phase. During the relaxation phase, here you are giving two successive stimulus. Same thing, here you are giving two successive stimulus. You can see here there is incomplete relaxation between two contractions. If you give same thing, if you give multiple number of time, what you will see? You will see that there is incomplete relaxation in between contraction. This is called the phenomena of clonus. Now, if you give the multiple stimulus during the phase of contraction or in the latent phase second part, what will happen? You will see there is summation of contraction in case of multiple stimulus. And there is a phenomena of titanization that will occur. Titanize. So you can see simple muscle twitch. This is simple muscle twitch. Here you can see there is summation of waves that is occurring. And here you can see relaxation incomplete in between. This is the clonus phenomena. And here you can see summation of contraction that has occurred. That is the phenomena of titanus. What we have told about staircase and tripping, you are giving just the stimulus just after relaxation. So what is happening? You can see one contraction, height of one contraction is increasing. The contractions are increasing gradually in height like a staircase. This is known as staircase phenomena or trippy. What is happening here? There is accumulation of calcium. Same beneficial effect is occurring here. Decrease in viscosity, increase in temperature in the muscle. In this picture, you can see that there is incomplete relaxation in between contraction, the phenomena of clonus or incomplete titanus. The skeletal muscle, the refractory period is short, so it can be titanized, while in the cardiac muscle have got long refractory period, so they cannot be fatigued or titanized. So your heart continues to beat without getting fatigued or titanized. So you can see summation of contraction. Ultimately, there is fatigue in the muscle, so here the graph is coming down. So this is simple muscle twitch. These are multiple stimulus given, which has resulted in titanization of the muscle. Same picture you can see here, titanus, incomplete titanus, clonus. Here you can see the staircase phenomena. These are effects of multiple stimuli, effect of multiple stimuli. You are increasing the, here say it was 1 per second. Here you have increased the frequency of stimulation to 5 per second. Here you have increased the frequency to 10 per second. Here you have increased the frequency to 50 per second. With that, the muscle is getting titanized. So fatigue. So after a long, it is the decrease in performance due to continuous and prolonged activity. In the CNS, the site of fatigue is the synapse in the new, and in the muscle, it is basically the neuromuscular junction. So if you give rest to the muscle, after some time, you will see the muscle will recover. Fatigue, why the fatigue occurs? You see, there are neurotransmitter vesicles that are released from the presynaptic terminal into the synaptic gland. After some time, there will be exhaustion of the neurotransmitter vesicle. There will be an ionic imbalance that is created in the muscle end plate as well as muscle end plate. And what will happen? As a result, there will be, the muscle will get the accumulation of hydrogen ions. All these will result in the phenomena of fatigue. If you give rest to the muscle, there is again regeneration of neurotransmitter vesicle. You supply nutrients, and finally the muscle is able to contract again after some rest. We have seen that free load or preload starts acting on the muscle before the muscle has begun its contraction, and afterload starts acting after the contraction, muscle has begun its contraction. We should know something called heat rigor, cold rigor, calcium rigor, and rigor mortis. These applied parts we will discuss in the next class. We will discuss about conductivity, tonicity of the muscle. The muscles, they remain in a partial state of contraction due to asynchronous gamma motor neuron discharge. This is called the tone of the muscle. Now, in upper motor neuron lesion and all, you can see there is some spasticity that develops in the muscle. And in the basal ganglia lesion, we will see there later on, there is rigidity, cogwheel and lead pipe type of rigidity that can develop in the muscle. Hypertrophy and hyperplasia of the muscle. These are the increase in size, that is hypertrophy, and hyperplasia is also increase in number of the muscle fibers. Now, age-related changes in skeletal muscle. Aging is associated with loss of muscle mass. Rate increases after the age of 50 years. Regular exercise training can improve strength and endurance of the muscle. Hypertrophy and hyperplasia of muscle can occur due to exercise. Say you have got a plaster in your hand, you have broken your hand, so your hand has been put on plaster. So after 6 weeks, your plaster is removed. You can see the hand which was in plaster is thinner than the hand due to which was active. This was due to disuse. There was some atrophy in the muscle. Now again you use it, you will see it has regained its bulk. The receptors in the muscle are the muscle spindle. They detect the change in length of the muscle and rate of change in length of the muscle. While the Golgi tendon organ, they detect the change in tension in the muscle. You can see the structure of the muscle spindle here. The muscle spindle, these are the receptors present in the muscle which detects the change in length of the muscle and rate of change in length of the muscle. These are the Golgi tendon organ which are activated when there is increase in tension in the muscle. So we will discuss about other properties and parts of the skeletal muscle in the next class. Stay safe. Bye.